In phylogenetic analysis, we have two things. Number one was evolutionary information and the other one was hereditary information. Evolution information is that thing which changes across different species. And on the other hand, hereditary information, it usually doesn't change across different species. ये hereditary information कैसे capture करते हैं? For that purpose, we have a concept that is called conserved regions. In conserved regions की base पे, based on proteins, we want to define three more concepts, which are basically our motif, then profile, and then domain. तो ये motif क्या है basically? Motif is a common concept across DNA, RNA, and protein structures, and basically it is a short conserved region in these three macromolecules which gives information about structure and function which are being performed by these macromolecules. So ये क्या है कि ये एक ऐसा हिस्सा किसी भी DNA, RNA और प्रोटीन का which captures or which carries structure or functional information. अब होता क्या है कि this information it should be shared amongst different sequences when they are aligned with the help of multiple sequence alignment techniques. और इनको रिप्रेजेंट कैसे किया जाता है विद द हेल्प ऑफ पोजीशन स्पेसिफिक मैट्रिसेस विच आर आल्सो स्कोरिंग मैट्रिसेस. अब ये मैट्रिसेस बेसिकली क्या हैं? वन ऑफ देम इज प्रोफाइल मैट्रिक्स. प्रोफाइल मैट्रिक्स क्या होती है? इट इज बेसिकली अ पोजीशन स्पेसिफिक मैट्रिक्स which gives us information about a motif. Commonly used for proteins, of course, that matrix is of size of 22 sequences, and what will happen? First 20 positions are for amino acids, and the remaining two positions are for gaps which can be there in amino acid sequences. So in profile, what we have among the rows of the profile, what we have our aligned amino acid sequences or residues which are there when we are aligning multiple sequences, and those residues which have the highest score, what they give us? They give us a consensus. What is consensus? Consensus is basically a voting where a higher number of votes give more accuracy. So then we have protein family concept. What is a protein family? Basically protein family consists of another concept, that is domain. Domain is roughly a part of protein. Domain is a part of protein, but it is usually greater than a motif. And what it has? It can fold or unfold, and it can carry more information than a motif. So protein family. Now what happens in protein family? This is set of different proteins which are interconnected or which have relationship between them. And then these members, they can be very similar, and then they can be very diversified or very dissimilar. So moving on, what is the purpose then of this information? Based on this information, we have different databases which are available for protein sequences. Among these databases, if we say, we have PFAM, blocks, prints, PROM, and PROCIT, and there is a common name for these databases also, which is called secondary databases. Why? Because they are not directly taken from protein sequences. What is happening? They are basically taken from primary databases, and then they are changed from those primary databases. How they are changed? As I said earlier, what we do, we find out conserved region from a given protein, and based on that conserved region, we make another database which is called secondary database. So finally, we have protein families. So as I said, protein families are collection of more than the two protein families, and then how they are made by making multiple sequence alignment, and which conservative fields are equal to each other or similar to each other, they are put in the same family. There are two tools then, PFARM A and PFARM B. So PFARM A is basically curated database of gap profiles. As I said earlier, that it is taken from initial or primary protein sequences, and the other one is PFARM B, which is generated automatically from sequences from the Proteome DB, and it usually has those information also which is not present in PFARM A. And finally, how we derive this information? With the help of hidden Markov models, which are basically again time serial models to have information which is based on likelihood of different protein sequences.